random and directionless. Yes, that is also correct. So yes, you guys remember genetic drift, change in frequency of an allele randomly. Yes, that is also correct. So yes, genetic drift is very clear. Now next is gene flow. What is gene flow? Change in frequency of a gene. Okay, due to a random event. Yes, Sampurna. That is also correct for genetic drift. So yeah, let us go to gene flow. What is gene flow? New generation, new population. Okay. Uh, okay, I didn't get this, but okay. Gene moving from one population to another and interbreed. Okay, yes, correct. Mutation, genetic changes brought by crossing over of alleles. Okay. Flow of genetic info from one population to other. Gorilla, yes, that's correct. Okay. So, yes. And yes, I hope you guys did see the assignment because I did remember that. And also, I saw the responses you gave for the first assignment. They were really amazing, I must say. Yes. Okay. So, yeah. So we are done with genes. So yes, mutation. Okay, okay, you're on the next question. Fine. Okay, so yeah, we'll go to mutation. Migration of organisms from one population to another, resulting in genetic variation in both the populations. Yes, that is correct. That is very appropriate. Yes. So that is very correct. Now let us go to mutation, the underlying principle of all these changes. Yeah, you already answered gene flow by mistake. Okay, Kaki, that's fine. Maybe that is the reason you jumped directly to mutation. Okay, so yes, single step and sudden. Yes. Okay. Random and direction, right? Okay. Yeah. Change in nucleic acid sequence. Yes. Correct. Okay. Mutation is a sudden change in gene. Yes. All of you are correct. Variety in traits. Okay. Yoga Devri's theory. Yes. It was the theory put up by Yoga Devri that mutation results in evolution. It is correct, Giveberry. Okay. Next question. Can you explain can you state the mating systems and how does parental care influence mating systems? Okay. Polyandry, polygamy. Okay. Monogamy. Yes. Polyandry, polygamy, monogamy, polygamy. Yes. Correct. Polygamy. Yes. And how does how does parental care influence mating systems? Okay, I know this will take some time to type, but yeah. When parents stay with their young and do not change partners is monogamy. Yes. Okay, correct. Depends on whether taking care of young ones is beneficial for the male or female. Yes, correct. Sampurna, you are correct. And partners are changed and the young are left after they are born leads to polygamy. Okay, that is also correct. Yes. So yes, you guys are correct. Maybe we'll go to the next question. What is sexual dimorphism? Morphological differences between the sexes of a species. Yes, correct. Male, female. Okay, what like difference is in male, female. Okay, yes. Difference in form between different sex and same species. Yes, that is also correct. Like that is correct. That is what it means. Yeah. So what is mate? Okay. Sex of same species but exhibit different character morphological. Yes, that is also correct. Yeah, coming to sexual dimorphism, can you tell like in polygyny like which one is more ornamented in polyandry, which one is more ornamented? And with whom does the choice lie? Who has the choice? Okay. In polyandry, female has the choice. Are you sure? Gynae females are and in polyandry males are. Yes. Equal ka ki, are you sure in polyandry female has the choice? Because in polyandry female is the one ornamented. Yeah, polygamy includes sexual dimorphism in size as well. Gynae females are. Yeah. Gaurija, you're correct. In gynae females have the choice. In andry, polyandry males have the choice. Correct. Okay. Yeah. Okay, now let us go to the next question. What is made choice? First of all, what is made choice? Actually, I should have asked the question later, right? In polyandry, who has the made choice? Okay, fine. Choosing who to breed with. Yeah, that is correct. Anyone else? Factors. Okay. Choosing a mate based choosing a mate based on their qualities, as in more desirable qualities. Yes, that is correct. Like choosing the good genes or something. Yes. Intersexual selection. Yes, that is correct. Physiology and anatomy. Okay. Wait. I'm asking as in like okay, so basically the female or like whoever has a choice will choose the other individual based on its characteristics, right? Yeah, the good gene hypothesis comes in here. Yes, that is correct. So what are the factors that will influence the choice? That's what I'm asking. Like physiology and anatomy is correct, but then yeah, okay. Where the highest chances of reproductive success are? Yes. Female peacock desire to mate with a mate who has colorful plumage. Yes, that is correct, to be very. Yeah. So yes, you guys remember everything. Now let us proceed to today's session. I think you guys remember I asked you a question last time. Like why is the behavior of worker bees favored? So let us go with it today. Okay, just a second. Sorry, I couldn't connect to you. Okay. Genetic traits that can enhance the survival and reproductive success of the offspring are favored. Yes. Okay. You couldn't connect to me as in I didn't get to you. You were very okay. So basically, okay. Last time I had asked a question in the last session that worker bees, they sacrifice themselves for the hive, right? Like they when the hive gets attacked, they basically sting the organism attacking, and in that way they themselves get killed. Now for this behavior, neither their individual success is favored nor the reproductive success is favored. So then why is this behavior favored? Yeah. Yes, I also gave an example of antelope alerting the rest of the herd, and it's like, yeah, it gets attacked because the lion gets attracted because of that call. Okay, so yeah, but then why is this behavior favored? That is the major thing that we will see today. Okay, so here we go in the third part of the quest of ethology. Okay, yeah. So yes, this is a quick recap of what we did. We actually did the recap through the review questions, but again, okay. So last time we did some basics about genetics. Yes, this is the last session. Everybody. This is the last session of the quest of ethology. This is the last session of the summer school as well. Okay. So yeah. So we got some basics about genetics. Then later on we went to some theories put forth about evolution. And then we went to the function, that is the individual success, the reproductive success, etc. Okay. Oh, okay. So yes, this is the question that I asked, that natural selection favors some behaviors which increase individual or reproductive success. Now, suppose a herd of deer is grazing. One of them sees a lion and gives an alarm call. Others run away, but the one which gave the alarm call gets hunted by the lion. Now, for this behavior, neither individual success nor reproductive success are favored. So then why, like, both of them are not enhanced. So then why is this behavior favored? And this is not only the one condition where such sacrificial behavior is observed. There are many examples. Like, this kind of behavior is called altruism. Like, you know, I am hungry, but I know you are hungrier. That is altruism. Basically, sacrificing yourself for some individual. So the unselfish behaviors, they are called as altruism. Is it for the greater good? Uh, no, not actually. Wait, we'll come to it. The question still remains, but we'll come to it. Don't worry. Okay. So the worker bee which sacrifices itself for the queen bee. Also, the picture which you can see, these are naked mole rats. They have like a similar thing like the honeybees. So the queen which is giving birth and the other, uh, what do we say? The other naked mole rats basically take care of the queen and her children. So these are some examples of unselfish behaviors like altruism. But the question still remains, right? Why is this behavior favored? So to answer this, Hamilton put up a concept which is called as genetic success. Now, as you can see, like genetic success, as in the genes of the organism are conserved somewhere. Like, suppose the herd of the deer, which I said was grazing. Now, the other deer which got saved shared some genes with the one who was killed. So, for the conservation of those genes, he sacrificed himself. Same is the condition with worker bees. Now, we all know worker bee and queen bee are like somewhat related, right? They are sister siblings. So, that so the some of the genetic content of the worker bee is present in the queen bee. For that genetic content, it sacrifices itself. So to sum all these things up, Hamilton put up a concept called inclusive fitness, like both the fitnesses. So in that inclusive fitness, he included two things. It is direct. It like the behavior should influence direct success as in individual success or reproductive success. The success of the individual. Okay. And indirect success, that is the genetic success. Okay. So now that I'm putting up genes over here, he also stated a way to somehow quantify it. Okay. So yes, Hamilton's rule. So Hamilton put up a rule to somehow quantify it. Okay. So basically, now let us see. This is the rule where R is the coefficient of relatedness, B is the benefit, C is the cost. Okay. And a certain behavior is favored if RB is greater than C. Okay, if I call RB as some value, maybe if I say that RB is a value, so that R into B, like the coefficient of relatedness into benefit, should be greater than the cost incurred. And if this relation follows, then the behavior is favored. This is what Hamilton stated. Okay. And now that you know the coefficient of relatedness comes into picture, that is, we are going to favor the kin, we are going to favor our family. So natural selection that favors altruism in this way, enhancing reproductive success or something. So that is called as kin selection because we are giving preference to the kin. Okay. Let us analyze this RB greater than C with an example. Okay. So this is an example. A person goes surfing with his sister. Okay. A person goes surfing with his sister. His sister saves him from drowning. He is about to drown and his sister saves him. So we will analyze this behavior. Okay. We consider that both of them are a part of average human population where one individual has two offspring. Okay. And also we have given that the probability of the sister drowning while saving him is 25%. Okay. Both of them can't drown since they are good swimmers. We assume that. Okay. We assume that both of them are good swimmers, both of them won't drown. And if the sister doesn't save this person will drown. So now we are left with two cases that the sister saves him or she drowns while saving him. Okay. Benefit of this act is the two offsprings of that individual. Okay. That is the benefit. Okay. So because if he would have drowned, if she wouldn't have saved him, he would have drowned and like two lives would have gone of the offspring of that individual. So that is the benefit incurred of this act because she saved him. So the benefit of this act becomes 2. So B is equal to 2. Okay. Now let us go to the second condition where we consider that she would have drowned while saving him. Now he got saved, she has drowned. So the cost. Now, okay. What is the probability of she getting drowned while saving him is 25%. That is 0.25. So that is the probability that she would have drowned. So if she would have drowned, then the cost would have been the 2 offsprings of the sister. So basically the entire cost thing would be 0.25 into 2 because the condition is if she drowns, right? Then the 2 offsprings will be the cost. So the entire cost will be 0.25 into 2, which is 0.5. Okay, I hope you all are with me till here. And R, that is, since they are real siblings, coefficient of relatedness, they share half of the genes with each other approximately. So we can assume that R is 0.5. Okay, so are you with me till here? Put a thumbs up if you are with me. Yes. Okay. So now we have the values of B, R, and C. So B was 2 because of the 2 offsprings of that individual getting saved. C was 0.25 into 2 because the probability of him drowning, the probability of her drowning into her 2 offspring. So that was 0.5. So we have B is equals to 2, C is equals to 0.25 into 2, which is 0.5, and R is 0.5. Now that we have all three values, let us analyze. So now R into B, that is 0.5 into 2, which is 1. Now 1 is greater than the cost. What was the cost? 0.5. Consider, suppose we say we consider we consider two populations. Okay. In one population, the sister saves him. In the other, the sister does not save him. So the population in which the sister saves, like this behavior will be favored, and the population in which the sister saves will grow more, right? So this behavior is basically beneficial for the individual as well as his sister. So this is how we can analyze a certain behavior with respect to RB greater than C. So okay, let us see first of all cost. How do we get the cost? So the cost is the probability of that individual getting risk or dying, that probability into the number of offsprings that person could have reared. That is the cost. The benefit is the offspring that will be reared by the individual saved. Okay. And R is the coefficient of relatedness, as in how much genes they share. Your video is rotated. Okay. If possible. Okay, fine. Is it okay now? I guess it is. Okay. So I hope you guys understood. Okay, for you guys to better understand, I have a question. Let us see if you guys understood this thing, if I was successful in explaining this thing to you. So yeah, just a second. Yes, face the case. Okay. So there is a population of rats in which one rat can have 5 offspring. In the natural environment, they give alarm calls on sight of the predator. One rat who has the probability 30% probability of getting caught after giving an alarm call. He gives alarm call to save his first cousin, where R is 0.125. So is this behavior favored? This is like mostly similar to the previous question. I have given all the values. Just analyze if this behavior is favored. Okay. Okay. So okay. Let us start from the beginning. First condition. What will be the benefit in this case? What will be B over here? First cousin being saved. Okay. So yeah, first cousin is being saved, but like I'm asking for the value. Like uh, B will be the value. Like after the first cousin gets saved. So like, what is the benefit of that cousin getting saved? 0.2 genetic success. Yes, the yeah the okay. See, the benefit of the cousin getting saved is genetic success, but like Hamilton said, all behaviors will not be favored. If RB is greater than C, then only the behavior will be favored. So I want you guys to see if in this case, which I have given you, is RB greater than C, is the behavior favored? Based on that, uh, what do we say? Based on Hamilton's rule, is this behavior favored? Consider this, okay, consider this as a hypothetical condition, okay. Of course, animals won't calculate and go to save, but consider it as a hypothetical case. So what will be the value of B over here? Okay, fine. We'll start. Okay, we'll start together. The value of B, now since the first cousin gets saved, assume that the first cousin gets saved. So the 5 offsprings reared by the first cousin will be the benefit, right? If the first cousin gets saved, she will be able to rear 5 offsprings. So the benefit is 5, right? Okay, are you here with me? Yeah, right. So B is 5. Yes, okay, you got it. Correct. So now what is the cost? What will be the cost? It's clear now. Okay, good. So now since it's clear, okay, can you please repeat how to get B? Okay. So see, if if the first cousin gets saved, yes, 1.5, that is correct. Cost is 1.5. So okay, if the first cousin gets saved, she will be able to rear, okay, he or she, okay, it will be able to rear 5 offsprings, right? So that is why the benefit of this act, like the rat giving the alarm call, the benefit will be 5. The 5 offspring of the first cousin. That will be the benefit. Okay? Is it clear with you, Malito Sugi? Yeah, 30% probability of getting caught. 1.5. Okay, Malito Sugi. Okay. So now that it's correct, now you can maybe go for calculating the cost. The rat has 30% probability of getting caught. And if he gets caught, the 5 offspring of the rat, which is getting caught, That will be the cost incurred. So what will be the cost? Okay, yes. And Rose, Roshan, yes. Yes. RB is 0.625, C is 1.5, yes. So no. Correct. This behavior is not favored. You are correct. Others, did you get with C? Are you guys okay? Are you guys there at C? Yeah. What's the equation for this? Okay, the equation is RB should be greater than C. That is R into benefit relate coefficient of relatedness into benefit. Yes, Gaurija, that's correct. RB should be greater than C. If this is satisfied, the behavior is favored. Okay, yes. And Jyotin, you're correct. Okay, how to get cost? Okay, so now the rat. Okay, and how many rats are being considered? How many rats as in? Okay, so no, I didn't get your question. How many rats are being considered? I didn't get that. Okay, so Malita Sugi, to get cost, yes, you're very cost, that is correct, 1.5. So to get cost, see, what will be the cost of this behavior? Okay, he gives an alarm call and he has a 30% probability of getting caught. Okay, and if he gets caught, the 5 offsprings that could have been reared by him, that will be the cost. Yes, and Jatin, that's correct. Cost is equals to probability of getting caught into offsprings per rat. Simply. But yes, that is the explanation behind that. Like if it gets caught, then the 5 offsprings. Like first of all, see, first condition is if it gets caught. So we need to consider the probability of that rat being caught. And if it gets caught, then the 5 offsprings that could have reared, that the rat could have reared, that is the cost. Okay, the fast rat first cousin 10 offspring, 5 from each. Yeah, got it. Thanks for explaining me. Okay, very good. So now that you are able to calculate the benefit, calculate the cost, now tell me if this behavior is favored. Yes, fast rat first cousin 10 offspring, 5 from each. Yes, correct. Yeah, so okay, I've already got one answer, but I'm waiting for others. Is this behavior favored? Now that you got B, you got C, you have R, so now calculate if it is favored. Not favored. Cost is greater than RB. No, it is not. Yes, Gaurisha Sampurna, yes. Rakhi Shailaja, yes, you're correct. It is still a bit fuzzy, but it's more clear now. Okay. No, here RB is greater than C. Okay. So, yeah, okay. I'll share some, maybe after this, I'll share some links where you can, understand this more clearly. Will that do? Ikoikaki? Okay. 5 into 0.125 is greater than 5 into 0.3. Is this how it is substituted? Yes. Yes, Malita Sugli, you're correct. So here you got it right. So B was 5, R was 0.125, and your entire cost thing was 0.3 into 5. So yes. Okay. Thank you. So yes. Now that you all understood, now we'll go ahead. This is how you can actually you can apply Hamilton's rule. And now when it comes to be like greater, it can be greater than 0.5. You can see that worker bee is the sister of the queen bee. So then because of the interbreeding and all these things, R happens to be greater than R can be greater than 0.5. But yeah, R has larger values in altruistic populations. That is what is studied. Okay. So yes, I hope this was interesting and you guys got how to you guys understood how to analyze this. Now let us go ahead. Okay. So yes, reciprocal altruism. Now as you can see, this okay. So, okay. What is the minimum value of R in all cases? See, the minimum value can be like very, very small, right? If you consider very less related species, like, you know, our relatives, dur ke rishtedar, so that can be very less. So R depends, like you what kind of relatives you are considering. But when it comes to like the populations, which I'm saying, the altruistic populations, there because of interbreeding or mating with your sisters, your siblings, and all these things, R happens to increase because of those things. It is greater than zero at least, right? Yes, of course. Otherwise, the equation is totally gone. Okay, so now let us go to reciprocal altruism. So as you can see, reciprocal altruism. So this creature which you can see, it's a bat. Okay, it's a vampire bat. The vampire bats feed on blood, and it is like if they don't get blood for 2 or 3 nights, they might die. So that is the condition. So what happens is if suppose one bat shares his blood to the other bat for one night, he expects that behavior to be reciprocated by the other bat sometime, someday. And if the other bat doesn't reciprocate this behavior, then that the other bat is boycotted. Like he's kind of considered betrayal. That is considered betrayal. So he is boycotted from the population and all those things. Like something similar to that. But then how to explain this? Cannibalism. No, no, not cannibalism. They feed on the blood of other organisms. Boycotted as in like he will no longer. Okay, no, no. So he will no longer be helped by the population. That is what it means. Do they feed? No, 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 no. They don't feed on the boycotted bat. They won't help him later on. Like they won't include him in the population or something like that. Basically, they won't help him later on. If he needs blood later, they won't help him. So that's what. Then they like scientists said that this is a tit for tat mechanism. Okay, it's just an outcast. Yes, similar, somewhat similar. Yes. So the scientists said that it is a tit for tat mechanism. Like suppose if two individuals or two species are interacting, so if their first interaction is altruistic, they will behave altruistically until one of them like betrays or doesn't behave altruistically. So that is the strategy used generally. That is what scientists have found. Okay. And now going to the second thing, Hamiltonian spite. Hamiltonian spite as in, I will kill you even if I will have to stay hungry. That is one line which describes Hamiltonian spite. Like you guys must have heard about infanticide, right? Yeah. Infanticide or lions killing the cubs. Yeah, so this is called as Hamiltonian spite. That yeah. So now yes, this is a question for you guys. I guess I'll put it in your assignment as well. Why is Hamiltonian spite favored? Like there is no individual success, there is no reproductive success, there is no success of the species. Why is this favored? And actually you are, killing your own offspring. So own offspring in the sense, yeah, you are killing the cubs. So then why is this favored? It is a question for you guys. Think about it. Of course, it will be a part of your assignment as well. Could you please explain again? Okay. Hamiltonian spite is like, it is the opposite of altruism. A symbiotic relationship is a form of altruism. Symbiotic relationship. No. See, in symbiotic relationships, the the individuals which are involved, the species which are involved, they gain some benefit with that thing, right? So that is not a form of altruism. You can't say that because first thing is symbiotic relationships. It is not a form of behavior. Altruism is a form of behavior. First thing, like it is a behavior. Okay, symbiotic relationship is like it it exists. Maybe the evolution of altruism and evolution of symbiotic relationships happened in the same way, but both of them are different. Okay, and coming to Hamiltonian spite. So Hamiltonian spite is like the opposite of altruism, where you must have heard about infanticide or lion killing the cubs. So these are the examples of altruism. Sorry, of Hamiltonian spite. It is like I will kill you even if I want to, even if I'll have to stay hungry. That is one line which can explain Hamiltonian spite. Okay. And this is a question for you guys. Think about it. Why is Hamiltonian spite favored? How is it favored? Why or how is it favored? Why does nature select Hamiltonian spite to be there? When there is no individual success, no reproductive success. Why not individual success? Hunger? No, no, no. Eat the, they won't eat the whatever, the other species. No. Infanticide, okay, lion, must have heard about lion killing the cubs, right? So when the lion kills the cubs, he won't eat it. He won't eat them. He kills them for a different reason. You can look up to it. Okay. You can look up, you'll get the answer. It's very interesting also. But this is a question for you. First, give it a thought. Why will it be favored? Okay. So, so altruism is selflessness and spite is selfishness. Yes, that is correct. Okay. So, like we somewhat have analyzed all the aspects of function. Now we'll look at some further advancements of genetics. Initially, I told you that Mendel put up very, what do we say, simple things, right? Dominant alleles, recessive alleles, all that. Maybe this is favored because the animal doesn't want to lose its place in hierarchy. Okay, hierarchy as in which hierarchy? Or it's removing offspring which is not fit. It actually doesn't select that way. See, you're thinking on the correct lines. It is related somewhat to the genes, but then that is one of the reasons when like if you consider the example of lions, yes, that is one of the examples. But yeah, there is some influence of the genes as well. You can look up to it. Okay. So now let us look at some further advancements in genetics. Okay. Since we will now be going to somewhat genes, so we need to know about some advancements in genetics, right? So initially, Rosie the cow. Oh, okay. So initially, when Mendel started the experiment, he said that one of the alleles will be dominant, one will be recessive. That the dominant allele will completely mask the effect of the recessive allele. Oh, okay, okay. Okay, so the dominant allele will completely mask the effect of the recessive allele. That is what Mendel said. What if this does not happen? What if the dominant is not able to be completely dominant? In those conditions, like the dominant is not completely dominant. So the effect in the first generation appears mixed. As you can see, red and white are caused and first generation are all pink. So the characters are blended, right? So that is called incomplete dominance. Here the characters are blended. Okay. Now either the characters get blended or they express themselves. Yes, that is seen in Snapdragon. You're correct. That is also seen in Snapdragon. So that is incomplete dominance. And when it comes to codominance, like both of them express themselves differently. Okay. So like as you can see this, if this white and red made 4 o'clock click plant, yes. 4 o'clock plant, yes. So okay. And as you can see here, so the white and the red, when they mate, it gives a roan cow. And then both of the characters are seen individually. So that is codominance. Okay. Pleiotropy examples, starch and seed size in pea plant. Okay. We'll come to it, Tikoikaki. Just a moment. Okay. So yeah, in the previous sessions, in the previous modules, okay, in one of the sessions taken by Samiksha, you guys were told an example of codominance. Can you name it? Samiksha did tell you an example of codominance. ABO, yes. Okay, so over here, which two are codominant? Gaurija, blood group, yes, correct. Blood group, correct. So here, like, what is okay? AB type, yes, correct. Rakhi said that. Coat color in cattle, yes. Coat color in cattle. That's what I'm showing. The roan cow. Yes, that is also correct. Blood group, nice. So AB type where A and B both the antigens are present on the surface of RBC. Yes. So okay. So you guys got codominance, I guess. So also, like initially when gene concept came into picture, what people thought was like one gene makes one protein. That is what they thought. But now studies show that maybe one gene or like one gene can regulate one trait. What if one gene regulates many traits? Both alleles are dominant. Yes, correct. Both the alleles are dominant. Like you can't say dominant, but then the recessive is not completely recessive and the dominant is not completely able to mask the effect of recessive. Hence, both of them appear. Yes, you're correct. Okay. So, incomplete dominance. Yeah, in incomplete dominance, the effect of both the alleles appears mixed. In codominance, both of them express independently. Okay. So now, if like we thought, yes, one of the alleles is dominant. So okay, we initially thought that one gene will regulate one trait. What if one gene regulates many traits? That is also a condition possible. Like Ikoikaki did give an example before. So yes, that is the condition is called as pleiotropy, where one gene regulates many traits. Yes, pleiotropy. You're correct, Yuvibari. So one gene regulates many traits. So yeah, there are many examples of this condition, like the one which Ikoikaki gave some time before. But yeah, there are many conditions. Sickle cell anemia, yes, that is a very nice example. Sickle cell anemia. Actually, sickle cell anemia explains many of these things, but we'll talk about it sometime. Yeah, Nilketonuria. Okay. So yeah, that is also correct. Also, like there is one gene in the pea plant which influences two, three traits. Like I guess it influences the pod color and it also causes a red spot at the exile of the plant. So yes, that is also a condition. The two, three traits which can be regulated by a single gene. Now let us consider the opposite case, where many genes regulate a single trait. That is called polygenic inheritance. And you can observe, like in polygenic inheritance, you can observe many phenotypes, as in like a variation, a lot of variation in the characters is observed in case of polygenic inheritance. So the examples of polygenic inheritance are kernel color in wheat, yes, skin color in humans, correct, UV Barry. Skin color in humans has a polygenic inheritance. You can see how much of variation exists in the skin color, right? So that is an example of polygenic inheritance. Next is multiple alleles. Till now, what like we saw that there were only two alleles. One was dominant, one was recessive. But what if in a population there exist more than two alleles of a single gene? That is also possible, and it is quantitative. Yes, it's quantitative. So that is called as multiple alleles. As you can see here, the coat color in rabbit. This has four alleles, right? It can either have like it can be black, it can be chinchilla, Himalayan, or albino. So it has four alleles, out of which three are dominant, one is recessive. Also, like Samiksha, like the example of ABO blood group system, right? It doesn't have only two alleles; it has three alleles. So that is also the case of multiple alleles. Now, the next condition is epistasis. Epistasis, as in where one gene influences the effect of another gene. Okay. So now this is an example. Okay. The rat which is seen, the mouse which is seen over here. Now, having brown color is the dominant trait, and having black color is the recessive trait. Okay. And there is one gene. So one gene basically regulates the color of pigment, and the other gene regulates whether pigmentation is allowed or not. Okay. So now you can see that there are two genes, the A gene and the C gene, right? So A basically regulates the color of the pigment, and C regulates whether pigmentation is allowed or not. So now as you can see in this specific rat, like capital A, small c genotype. In this condition, or maybe capital A, small a, or all the genotypes, you can see the genotypes where A has like the rat has a pigment. Okay. The gene for pigment is dominant or recessive, but the pigment is present. Okay. Since the second gene is recessive, that is, pigmentation is not allowed, even when the pigment is present, the rat will be albino. Okay. You can see all these four cases. The white, the albino rat. The pigment is present, right? But even when it is present, pigmentation is not observed. Why? Because the second gene which allows pigmentation, that is recessive. So basically the gene which allows pigmentation, it affects, like it influences the effect of the gene which makes pigment. That is called as epistasis. Okay, did you get that? Like the effect of one gene is enhanced or suppressed by another gene. Okay, is it clear? Are you here with me till now? Give me a thumbs up if you are with me. Yes, clear. Okay, thank you. Yes. Okay. So now again, I have a question which you guys already answered, but yeah, I would like to ask again. Phenylketonuria is a genetic disorder, right? I get to know about this. Oh, Yeah, actually you told before UV Barry, but yeah, to know about this first time. Oh, okay. Actually, this is very interesting epistasis. You can look up on it. It has different cases, like supplementary genes and all those things. You can actually look up. It's interesting. Yeah, like he did answer, but then again, phenylketonuria, it is a genetic disorder. Like I guess it is because of a point mutation, and it occurs in. You can see that the gene is defective for the enzyme phenylalanine hydrolase. So that gene, it also causes mental retardation. Yeah, hyperpigmentation. Yes, yes, Sampurna, you're correct. Pigmentation in hair and skin of the individual. Yes, and Roshan. Yes, you're also correct. You be very yes. You did tell it before, but yes, you're correct. Okay, so yeah, I hope you guys were able to analyze this case. Or is it my network issue that I'm not getting the response? Okay, so yes, so that is what it is. Now let us go to something different. Again, one more question for you guys. Yes. Do the genes present only in the nucleus regulate traits in an organism? Like, okay, so till now what we saw, the genes are present in the nucleus. The DNA in the nucleus, it regulates all the traits. So is it that only that DNA will, show regulate the traits which are present in the organism? Do you think? Mitochondria has its own DNA. Will it affect anything? Okay, so what do you think? Will it affect? You're thinking on the correct line. Do you think that will affect? Like it is so small compared to the nuclear DNA, right? Nuclear genome is like okay, but it is very small. But will it affect? Yes. Okay. Can you give any examples where mitochondrial, like a certain trait is caused because of mitochondrial DNA? Control by RNA. Okay. Yes. Okay. There are other factors which can control the expression of gene. Okay, you were agreeing with me. Yeah, there are many factors which control the expression of gene. It's not only the gene itself. Yeah, lack of iron. Those are like that is a part of the nucleate of the bacteria, right? It is a part of the genetic material of the bacteria. Okay, so yeah, you were thinking on the correct lines. Mitochondria has its own DNA, and there are some traits which are regulated by the mitochondrial DNA. The example is Liber hereditary optic neuropathy. This is a disorder which is caused because of the like mitochondrial DNA. Plasmid. Okay. Do we have plasmid? Yes. So the no, not in us. Yes. So then like, okay, in bacteria, you can say that there are some traits. What was that disorder? Okay, it is Leber hereditary optic neuropathy. Wait, I'll put it. I was just trying to think of some examples. Okay, the optic neuropathy. Yeah, this is a very famous example used for mitochondrial inheritance. Leber hereditary optic neuropathy. It is a mitochondrial disorder, as in it is regulated by the mitochondrial gene. And like when we study human genetics, na, this is also a type of inheritance which is observed. Because since like you can, you know that the sperm is very small, right? It can give only the nucleus. The entire cell is the egg cell now. So the mitochondrial genes are from the mother itself. Okay, so basically this inheritance follows like from the mother to all her offspring since they have the same mitochondrial DNA. Okay, so this is also an example of mitochondrial inheritance. Okay. Oh wow. Yeah. Okay. So now, okay. Hmm. So yes, like UV Barry was stating, like RNA can control DNA. Are there more cases like LHON? Okay, so actually there were studies going on to see if diabetes is mitochondrial or not. I don't know if they have completed, but yes, like that is one example that I know. Diabetes can be mitochondrial, I guess. You can look up on it. Mitochondrial inheritance, like it is a type of inheritance. Yes. Okay. So now that you know, if a gene changes, the effect of the gene changes, right? If there's a mutation in the gene, the effect will be like the effect will be changed. But what if the gene is not changed, but still the effect is different? Okay. So the gene is the same, but there are some other factors because of which the effect of the gene shown is different. Like the character is altered. Okay. So these traits are called, these are heritable because like, okay. So you guys must have heard about, okay. So the study of these traits, basically the study of these genes and this trait is called as epigenetics. As in like, okay. So these traits mostly in the gene expression. Now, as Aryan also told you, like, you know, the transcription, translation, and all these things. So, when a gene is getting expressed, so you need to regulate it at various levels, right? So, these changes, like, which are the epigenetic changes, they mostly are in this regulation thing, the regulation of gene expression. So, the gene sequence is not altered, but the expression is altered. Okay. So examples are histone modification or DNA methylation, repressor proteins, enhancer proteins. Yes. Gene regulation. Okay, yeah. Gene regulation, correct. So the examples are histone modification, DNA methylation, etc. Repressor proteins, enhancer proteins, and these epigenetic changes, gene expression. Yes. So these epigenetic things, these epigenetic changes, sorry to interrupt, but would Jacob disease would be considered as well? This prions are the, you know, what do we say? They are the previous topic. Yes, prions are like the like epigenetic. You're asking, right? Can it be epigenetic? Yeah, prions are like rowdy proteins. Yes, you're correct. So prions are the proteins which are not folded properly, and hence they affect the like the protein is not folded properly. The problem is in the conformation of the protein. It is not the part of the gene. Yeah. So it is the part of the protein. It is not a part of the gene. I'm talking where okay, so the protein is made the protein is made properly, but its folding is changed. I'm talking here that the protein is only not made properly. Are you getting that? Okay, so like when I yeah, genetic material specifically, got it. So like see, suppose the DNA gets methylated. So it will somewhere alter the transcription, it will somewhere alter the translation, right? Because the DNA gets methylated. Or okay, repressor proteins you must have heard. Repressor enhancers. So repressor proteins, what they do is they basically block the way of the polymerase, right? So the gene is not changed. The gene is not altered, but the effect is altered. That is what I'm talking about. And since those are proteins, they will be passed on from generations to generations. So that is what is called as epigenetics. Okay, it is a very nice field of genetics. It is a part of genetics, and like due to non-genetic part, the genes behave differently. Okay, so now that we got some, okay, huh? Yeah, here again a question for you. Yeah, okay. So can you name an arrangement of several genes with a promoter and operator? I will just say lacoperon again. Yes, actually you're correct, Ikoikaki. Lacoperon. Is only does operon exist only for the lactose thing? TRP operon. Okay, correct. So like yeah, this arrangement of this thing is called as operon. So based on what that operon metabolizes, there are different names like lactose metabolizing operon is lac operon. Tryptophan metabolizing operon is called TRP operon. Yes, lac operon. Correct. So yes, you guys know lac operon and like you guys are pretty much yeah. So let us go ahead now, bringing back this gene to our behavior. So this is a field called behavioral genetics. It was there in NCRT. Yeah. Yes, lack of Oberon is there in NCRT. Yeah. So yes, and Oberon is a very nice, like it is very nicely explained in NCRT, I guess. Is it? Yeah. So yes. So now we go like we combine behavior and genes together. What comes as behavioral genetics? As in like studying the genes which influence the behavior. Okay. So it proc gene. Okay, what is it? Like I have no idea what is it. Can you please tell? Okay, so okay, I guess you're trying to say that it is in the prokaryotic genome. Yes, it is in the prokaryotic genome. Yes, so operons exist in the prokaryotic genome. In eukaryotes, like we have several other mechanisms of regulation. So yeah. So now, okay, when I say I combine behavior and genetics, so it comes from the concept of nature and nurture. Nature and nurture, as in nature is the genetic influence over something. Nurture is the environmental influence over something. Okay, so that is what is nature and nurture. So how do we study behavioral genetics? So okay, in case of animals, these are the methods of studying. Okay, so in case of animals, what we do is we try to like we carry out mating between siblings. over generations so that we obtain pure breeds. Now, as you can see, if you kind of, what do we say? If you self breed or interbreed these animals, siblings, so a lot more content gets shared between them. And based on that, now like we can maybe give them some different environments and study the effect of environment on them or study the effect of genes. Okay. Or maybe keep them in a similar environment and study the effect of genes. And then the other thing is selective breeding. Selective breeding as in I will try to mate the extremes, like the extremes of behaviors over several generations, and I observe how do genes get involved in these, you know, how can genes be involved in the behavior? Okay. So I like okay. So this is how it is studied in animals. When it comes to humans, when I want to study behavior genetics in humans, there are also methods to study. So the first method is twin method. Okay. So yeah, you guys know about monozygotic and dizygotic twins? I hope you guys know. Yeah. Okay. So yeah, you guys have an idea of what monozygotic and dizygotic twins are. So now that monozygotic twins share all their genes in common, but not when it comes to dizygotic genes, they are just like two different organisms born at the same time, isn't it? So the behavioral trait of dizygotic twins. Now, what? Yeah, identical and fraternal. Yes. So fraternal twins. So yeah, when I say about dizygotic twins, those are just two different organisms or like two different babies born at the same time. Okay, they have different genes because they are formed from two different zygotes, but they are like the environment in which they are reared by the same parents at the same time, same place. So the environmental conditions are same for both of them. So now their behavioral traits, like we can study now that they are now that they are in identical environment, we can actually study the influence of genes on their behavior. Okay, because the environment is same. So basically, I want to study the effect of gene and environment on behavior. I'm keeping one of them constant, so the environment is constant. So now I can study the effect of genes on their behavior. That is the twin method. Monozygotic twins share 100% DNA. Yeah, because they actually, it is like in case of monozygotic twins, what happens is in I guess 8 or 16 days of fertilization, the zygote splits into two parts, and both of them grow as different babies. So they actually come from the same zygote. So yeah, you can say that they share 100% DNA, unless there are some mutations. But mostly, yeah, they share 100% DNA. Hence, also like as Samiksha said, like rejection risk is less in monozygotic twins, least in monozygotic twins because they have the same DNA. So yes, that is the twin method which can be used. Second method is the adoption method. Like okay, this is somewhat difficult. Single fertilized egg split. Yes, single fertilized egg split in case of monozygotic. In case of dizygotic, two different eggs are fertilized. Okay, so in case of adoption method, what happens is children are adopted in early stages of life. So uh, it is like okay, this is somewhat yeah. So basically, what happens is the behavioral traits. Now I want to study the effect of nurture. So I will change the environment and keep the gene constant. So basically, I will study the effect of adoption. You know, the nurturing, the behavioral traits of the adopted mother and the biological mother. So like basically that is the condition where I can study the effect of environment. Okay. In the adoption method. Okay. So and why do we need to study behavior genetics? we can actually, the effect of studying behavioral genetics, or these studies help us improve understanding common uh, complex human behavioral disorders. There are many human behavior. Now that mental health awareness is increasing, there are many human behavior related disorders, right? So the analysis of those disorders based on genes can be done better using these analysis. Okay. So yeah, I will also share some links if you guys want to read more about it. It is a very interesting field nowadays. And yeah, so this is about behavior genetics. Simply the genes which regulate a certain behavior. Okay. So now, yeah, I hope you guys understood. Are you guys with me till now? Yes. Okay. So yeah, now let us go to, now let us, like, initially we combined behavior and genetics. Now let us combine behavior and epigenetics. Okay, but why is it only dizygotic and not monozygotic? Okay, so see, what the point is, I want to study the effect of I want to study the effect of genes. Okay, if I consider monozygotic twins, they will have the genes also same, the environment also same. So then, how will I be able to analyze that a certain behavior is like how much a certain behavior is influenced by the environment, how much by the genes? I won't be able to do that, right? Since dizygotic twins are two different zygotes but reared at the same time. That is why it makes me easier to study. Okay. So yes, now let us go to behavioral epigenetics, like combining behavior and genetics. So basically, without alterations in genes, when I change the gene and it affects the behavior of a certain organism, that is what is behavioral epigenetics. As in, like, suppose there is a certain incident in the life of an organism which causes, okay, that causes some epigenetic changes, and those epigenetic changes affect the behavior. So basically the analysis of these epigenetic changes which affect the behavior of the organism is called behavioral epigenetics. Okay, I hope you guys got this. Let us see it with an example. Okay, so okay, so these epigenetic changes, when I say, they are induced by experiences of certain trauma or stress, and those epigenetic changes change the behavior. Okay, so okay, let us consider a case. So some kind of childhood trauma. Okay, like suppose like maternal separation or childhood abuse. What it causes is, like, as I told you before, DNA methylation is a condition of epigenetic change. Okay, so what it causes is this childhood trauma. There's a axis in the brain, hypothalamic pituitary adrenal, which regulates our stress or something like that. Okay, it is the stress response axis, which uh, which creates a response to the stress. And because of this trauma, because of these experience, what happens is the working of that stress response axis gets somewhat disturbed. Okay, the genes are not altered. The genes are methylated, because of which the activity of that axis is altered, it is disturbed. And in turn, because the activity of that axis is disturbed, it increases the risk of suicide in that organism. It affects the behavior of that organism. As you can see, those epigenetic modifications, they can cause aggressive behavior, depression, or increased risk of suicide in organisms. So yeah, these studies are conducted on rats, but uh, yeah, but these things exist. So affect the behavior. Okay, it is simply that some epigenetic changes altering the behavior. Okay, is it clear? Are you guys with me? You give me a thumbs up if you guys are with me. Yes. Okay. So yes, thank you. Yes, even I think it is very fascinating and you know a very interesting thing to know that without even changing the genes, you can alter the if you can alter the effect, and that effect can in turn change the behavior. So yes, now we come to the end. So we identified the genes that can regulate a certain behavior. Now you can like just think about it, give it a thought. You can actually change those genes, right? So you can alter the behavior of an individual. You can make it the way you want by genetic engineering, of course. So just give it a thought. Where are we heading? Okay. So okay. Yeah, and yes, that is the end. Just yeah. So okay. As you know, Lord Buddha said, our thoughts drive our actions. So our behavior defines us, right? So yes, I hope you guys enjoyed this quest of ethology and you also got some many new things to learn. So yes, behavior is something we define our lives with. So it is always important to study animal behavior because it helps us study human behavior. And yes, so our actions define us, right? So yes, that is the end from my side. Signing off. Very goodbye. It was very nice interacting with you guys. You are the most interactive audience I've ever had. And yeah, it like I even enjoyed talking to you as well. So that is the end from my side. I am signing off. Thank you so much. I'm very chill. Okay, thank you. Okay, so even I'll okay, thanks a lot. Thank you so much. Okay, so yes, that is the end from my side. Even our other, even your other, even my other friends are in Samiksha. Even they would like to tell you something. Yes. What if we want to talk? Okay. What if we want to talk to you guys some other time? Yeah, you can always reach out to us. Like, okay, you. Okay. So you have our mail. You can always uh, come up to us. You can always talk to us. And also, if you guys want, we can give you our WhatsApp number. Yeah, thank you for explaining and repeating whenever asked. Okay, thank you so much. Yeah, it means a lot to hear all these things from you guys. Like how to get to know about future sessions and projects. Okay, so yeah, if you want to contact us, you can always mail us or you can ask questions on WhatsApp as well. Since you're in the like we all are present in the WhatsApp group, so you can talk to us as well. Yeah, it's fine. It's fine. I hope now that you are not confused and all the things I told you got them. So yes, all the other friends are here which took the previous module. They want to speak something. Yes.
Oh, okay. Hello, guys. This is Aryan again. So it was pretty great to see like such an interactive audience, and the fact that you've been there for like all 9 sessions. Some of you I recognize the names. So it was pretty great. So I hope that you really enjoyed our sessions. I hope you loved them because it was pretty good for us to also hold them. So just good luck in general for ahead. So yeah, that's it from me.
Hello everyone. I hope you enjoyed this summer school, and I would like to thank everyone for attending all the sessions and being interactive as well, because that motivated us to give our best. And all the best for your future adventures as well. That's it from my side. Thank you.
Hi guys, this is Ishwar. We'll sort out all your attendance and stuff within like 1 or 2 weeks. So we have some edge cases now. If some guys had a genuine reason and couldn't attend all the 6 sessions, like if you just attended 5 sessions or something because of genuine reason, just text us in the WhatsApp. And there are also few people who joined with a number instead of their name, like 123, like some random number was there. Those people have attended few sessions, so those people also just text us in the WhatsApp if you don't want to miss out on certification. And yeah, we'll just sort all this stuff out. And thank you for showing up every day, keeping the attendance high, and good luck on your future endeavors. If you want, just mail us to Biotech Club if you want to know anything or collaborate with us in the future. Thank you, guys.
Vaishnavi, you're muted.
Vaishnavi, unmute.
Oh, sorry. Okay, sorry. So yeah, I wanted to say that like we also got to know many things while teaching to you, and like while teaching you, we got to know many new concepts. We got to learn many new things. So even yeah, that was also a thing. And yes, you guys are really very awesome. Thank you so much. When will you all come up with next knowledgeable sessions? Okay, yeah, we'll let you know when we come up with those sessions again. Only WhatsApp channel is there, right? Is there yeah, it is there, WhatsApp group. Okay, fine. We'll put up a message on the WhatsApp group. Okay. Yeah. Yes. Thank you so much. And yes, a reminder today. Okay, fine. I'll put up my number as well. So yes, today is the deadline for assignment 6, and today I'll be giving assignment 7. So yeah, please note the deadline and let me know. Yeah, see, honestly, to say I've seen all your responses and you guys are pretty great. It's going to be difficult for me grading your assignments. But yeah, it is like mostly uh, it's fine. We'll like there's no a certain we haven't decided a certain pattern in which we will grade it because all our assignments were in different patterns. But yeah, like we will let you know about the assignments, attendance, and all those criteria through a mail, right?
All the
Yeah.
best to all of you, and I am glad I could participate in all these sessions. They were actually, worth our time. You guys put in a lot of effort. That was very clear. Yeah, I'm just really happy. Yeah.
Thank you so much. That means a lot to us.
Thank you, and
Yeah, I
also
Oh,
would.
thanks.
ayo.
Yeah, I
Thanks for
would
being.
unmute more, but I was confused, like if I should just use the chat or not.
Okay, so yeah, like you guys can stay back if you have any doubts, or yeah, you want to unmute and say something. That is also fine. Like that is also nice. Okay. So yes, and that's it from our side. Thank you so much, and have a nice day, all of you.
You explained about some projects in your session, Arin. So, are there any new projects now or still the old ones?
Oh no, just the 6 ones that we had explained last time. Yeah, those ones only.